To identify the 4 types of teeth and count their numbers in your mouth, stand in front of the mirror and open your mouth wide. Touch your teeth and count them. You must wash your hands before you touch your teeth. Try to identify the 4 types of teeth according to their shapes. At your age, you should have the following teeth according to the formula. Half of each jaw is equal to incisors 2, canine 1, premolar 2, molar 2 is equal to 7. To show that saliva breaks down starch to maltose, a sugar, take some boiled rice and divide it into 2 parts. Place them in 2 bowls, A and B, separately. Make a paste of the rice in bowl A and mix with 1 ml of water to it. Add a few drops of iodine to it. The rice turns blue-black in color, showing the presence of starch in it. Now take the boiled rice from bowl B and chew it well with the mouth. The boiled rice tastes sweet. Then take the paste into the bowl again, add 2 ml of water and then add a few drops of iodine solution. The paste will no longer change into blue-black color. When boiled rice was chewed, the salivary amylase of the saliva changed the starch to maltose. Thus it tasted sweet and gave a negative result with iodine. To draw and label the different parts of the human digestive system. Your teacher will bring a model of human digestive system and explain the parts. You will draw and label the diagram in your notebook. To show that water or moisture is lost during breathing, gently breathe out on a clean mirror and observe the droplets of water on it. Where did these water droplets come from? Exhaled air is warmer, and when this warm air touches the cold surface of the mirror, the moisture of the exhaled air condenses to form water droplets. To demonstrate that expired air or exhaled air contains carbon dioxide, take a test tube half filled with lime water. Inhale a lot of air and then forcibly breathe out into the test tube. You will see that the lime water turns milky. This shows that exhaled air contains carbon dioxide. To demonstrate the role of diaphragm in breathing, take a bell jar and fix a 1-holed cork onto its mouth. Insert the long arm of a Y-shaped glass tube into the hole through the other end of the bell jar. Attach 2 deflated balloons at the smaller free ends of this tube. Stretch a rubber sheet and tie it at the open end of the bell jar. In this model of the respiratory system, the rubber sheet represents the diaphragm, balloons represent the lungs, bell jar represents the thoracic cavity, Y-shaped tubes represent the trachea and bronchi. Now pull the rubber sheet downward. It will be seen that the balloons are inflated, representing inhalation. When the sheet is released, the balloons are deflated, representing exhalation.
The different types of blood cells in a permanent slide of human blood. Your teacher will take a permanent slide of the blood smear of human and focus it under the high power of a compound microscope. The numerous enucleated circular disc-shaped cells that can be seen are the red blood cells or erythrocytes. White blood cells can also be seen because of their lobed nucleus. But it is difficult to see the platelets even under the high power of the microscope. Draw the diagram of the blood smear of human showing the different types of cells. Find out your own blood group. Take 3 chemicals, antibodies, anti-A, anti-B, and anti-D. First, clean the tip of your finger with ether and then prick with a disposable needle. Take a clean slide and place 3 drops of blood on it at 3 different positions. Add 1 drop of the chemical anti-A to the first drop of blood, 1 drop of anti-B to the second drop of blood, and 1 drop of anti-D to the third drop. Take 3 clean toothpicks. Mix the chemical and the blood well at the 3 positions, each with a toothpick. Observe the blood drops carefully. After some time, clumping will be seen. If clumping occurs with anti-A and anti-D, then blood group is A positive. If clumping occurs with anti-A only, then blood group is A negative. If clumping occurs with anti-B and anti-D, then blood group is B positive. If clumping occurs with anti-B only, then blood group is B negative. If clumping occurs with anti-A and anti-B, then blood group is AB negative. If clumping occurs with anti-A, anti-B and anti-D, then blood group is AB positive. If clumping occur with anti-D only, then blood group is O positive. If clumping occur with none, then blood group is O negative. Collect blood for blood testing. Blood collection is a vital procedure in blood testing. In order to learn the steps of blood collection, storing and preserving, you need to visit a pathological laboratory. In the lab, the instruments needed for the blood collection and storing can be seen. It includes sterilized disposable needle, cotton wool, spirit for cleaning, plastic vials with caps for storing and tubes. An anticoagulant is added to the vials to prevent the clotting of blood. In cases where large amount of blood is needed, it is usually taken from the vein in the arm. But in the case where few drops are needed, it is taken by puncturing the fingertip. Doubt how blood tests can reveal the presence of infections and other diseases. If we go to a doctor complaining of certain difficulty in our body, the doctor will prescribe a number of blood tests in order to diagnose our disease correctly. The common blood tests done in a pathology lab and the diseases that they help to diagnose are enlisted below. Common blood tests, disease for which it is done. CBC or complete blood count helps to detect anemia, infections, clotting problems. RBC count, anemia, dehydration. WBC count, TLC count, immune system disorders, blood cancer, sign of infection. Platelet count, bleeding disorder or thrombic disorder. Hemoglobin percentage, anemia, sickle cell anemia, thalassemia. Hematocrit value, to identify anemia or polycythemia. Blood glucose level, diabetes, blood calcium level, kidney problems, bone disorder, thyroid disease. ESR, erythrocyte sedimentation rate, tuberculosis, rheumatic fever. ELISA test, detecting viral infection, dengue, HIV, etc. The pulse rate. Place the middle finger and the index finger of the right hand on the radial artery present on the inner side of the wrist of your left hand. If the fingers are pressed down, the heartbeat can be felt. Note the time and count the heartbeat. It will be seen that in 1 minute, 72 beats can be felt. This is the pulse rate. To note the difference in pulse rate or heartbeat under different conditions. After you have learned how to count the pulse rate, set different conditions and see how your pulse rate varies. Make a table with your findings. Condition: number of beats per minute. Remarks: jogging for 5 minutes, running for 2 minutes, lying down